Welcome everyone to this training. My name is Emmanuel Ezenwane. This training is about titration, and this video covers introduction to titration. Let's begin. What is titration?
Titration is a laboratory technique that involves slow addition of one solution of a known concentration into a known volume of another solution of unknown concentration. Therefore, titration involves two solutions. Let's say solution A with known concentration and solution B with unknown concentration. So with titration, the concentration of solution B can be determined by slow addition of solution A into solution B until end of reaction.
The end of reaction is indicated by color change, and it is referred to as end point. This titration is also known as volumetric analysis. Application of titration in chemical industries. In different chemical industries, titration is used to determine the unknown concentration of a solution. It is applied in different chemical industries such as pharmaceutical, food, cosmetic, and in research. Types of titration. We have acid-base titration, redox titration, precipitation titration, and complexometric titration. All these types of titration involve same technique, which we shall learn in this training. SSCE focuses on acid-base titration and redox titration only. We start with acid-base titration. This involves the use of titration to determine concentration of acid or base. In this type of titration, if the concentration of acid is known, then you will be using titration to determine concentration of base and vice versa. So it means that if the concentration of acid is known, then the concentration of base is unknown, and vice versa. To understand titration better, there are topics we ought to have covered. The topics include acid, definition and examples, strength of acid. There you'll be talking about strong and weak acids, base, definition and examples, alkali. We're talking about strong and weak alkali. Also, choice of indicators, when to use a particular indicator. At this point, you have to pause this video and take some minutes and revise these topics. Now you can continue. Terms used in titration. You have the term called titer. It is the minimum volume of solution added from the burette to reach the endpoint. Endpoint. It is the point at which the indicator changes color. Indicator. It is a substance that changes color when added into a solution depending upon the pH of the solution. Titrant is a solution with known concentration. It is contained in the burette. Analyte is a solution with unknown concentration. It is contained in the conical flask. Apparatus needed for titration. We have pipettes, pipette filler, conical flask, burette, and retort stand. Let's take a look at them. This is pipette, used to accurately measure a known volume of a solution. This is pipette filler of different sizes, used to fill up the pipettes. This is conical flask. It contains the liquid from the pipettes. It serves as the reaction system. This is burette, to add small measured volumes of one reactant into the other reactants in the conical flask. This is retort stand, used to hold burette. Titration setup. In the lab, this is how you set up the titration apparatus. Retort stand holding the burette against the conical flask. Let's look at precautions taken while carrying out titration. 1. Rinse burette with the solution of acid. Rinse pipettes with the solution of base. Remove air bubbles from burette and pipettes. Avoid splashes of solution on the walls of conical flask. Remove the funnel before titration. Very, very important. Most students do not obey this particular precaution, removing the funnel before titration. Always remember to remove the funnel before titration. Do not blow out the remaining drop of solution from the tip of pipette. Read the burette at eye level, or avoid parallax error. Use 2 or 3 drops of indicator. Avoid leakage of burette. Place the conical flask on a white tile or paper. We have come to the end of part 1. I look forward to seeing you in part 2. Thank you.